The video today is on membranes, or as I like to call them, biomembranes. When I'm talking about biomembranes, I'm talking about a phospholipid bilayer with embedded proteins. So, important things to know about biomembranes is they surround all cells. And we're talking about external membranes that surround all cells. All living organisms have biomembranes on the outside. And then some living organisms create membranes internally, and they make up organelles. So in eukaryotic organisms, we have organelles, which are made up of internal membranes, creating compartments and compartmentalization that allows for specialization inside the cell, right? These internal membranes, like I said, do a couple things. One, they compartmentalize. Right? So they allow you to have specific places for different metabolic processes. And by that, I mean like chains of related chemical reactions. For example, right, in eukaryotic organisms that do photosynthesis, all of those chemical reactions for photosynthesis happen inside the chloroplast. In eukaryotic organisms that do cellular respiration, everything but the first step of cellular respiration happens inside the mitochondria. That doesn't mean that these metabolic processes, photosynthesis and respiration, can't happen in prokaryotes like bacteria. They do, but they just happen on their cell membrane, not on internal membranes. So this compartmentalization is important for kind of refining and creating space for different reactions to occur in different locations. Also, these internal membranes help us increase our surface area for these reactions, which is incredibly important for increasing efficiency and productivity. So what are some of the parts of this biomembrane? Let's talk about that. So when we talk about parts of the membrane, the first most important part we want to talk about is a phospholipid. And as we know, phospholipids fall into our lipid class of macromolecules because they have a phosphate head attached to a glycerol with two fatty acid tails. Okay, they form the main barrier of the membrane. So they create the difference between the inside and the outside of the cell. They create that barrier that allows that difference to occur. Okay, so they form the main barrier of the membrane, and to separate inside from outside of the cell. So without the phospholipid bilayer, you wouldn't have a difference between inside and outside of the cell. So the cell couldn't be different than the environment. And that's the whole point of a cell, to have different homeostatic mechanisms than outside of the cell. And we know from before, like we said, right, we have the phosphate and glycerol head and then the fatty acid tails. So let's take a second and label those right up here. So here is our phospholipid bilayer, right? These are all phospholipids. Here's our bilayer. It's called a bilayer, right? Because we have two phospholipids facing each other. Here's a phospholipid right here. And here's another phospholipid right here. And again, why did they orient themselves that way? It's because they have polar and nonpolar regions, right? The head of the phospholipid is a polar head, right? And the tails of the phospholipid are nonpolar. What that does is that allows the phospholipids to face each other, and the outside layer of the phospholipid that's facing the external environment is hydrophilic. which means it doesn't mind being near all the water on the outside of the cell, right? And then again, on the inside part of the cell membrane, we again have these polar phosphate heads. So we have another hydrophilic region, which is important because inside the cell, we also have a lot of water. So now the phospholipids have oriented themselves so the heads are towards water, and the hydrophobic tails, right, this region right here, these hydrophobic tails are away from the water. So again, the main part of the phospholipid is to form the barrier. Now, what if it gets too cold? Okay, we want to prevent the phospholipid bilayer from freezing solid. So we put these little ring structures in here, 4 rings with a little tail, and those are our cholesterol. And cholesterol, these are another type of lipid, right? So let's go look down here at cholesterol. Cholesterol, again, fits in the lipid category because it is hydrophobic, but it's not made up of fatty acids. Okay, so the cholesterol are found between the fatty acid tails of the phospholipids, and it keeps the tails moving and fluid. Okay, so it helps prevent the phospholipids bilayer from fusing solid. It keeps them moving and fluid. And the amount is variable depending on the type of organism and different environmental conditions. So before bears go to hibernate, they actually add more cholesterol into their cell membrane to keep their cell membrane fluid as they get colder. Okay, so carbohydrates we're going to get to in just a second, but I want to at least talk about some of these proteins first. These proteins are incredibly important. Proteins have a diverse type and varied functions in the cell membrane. Okay, so transport proteins, we'll talk about this more when we get to passive and active transport, but we can have channel, we can have carrier proteins, we can have channel proteins, we can have protein pumps. These first 2 are types of passive transport proteins, and this is an active transport protein. And these transport proteins allow large and or charged molecules, and sometimes charged ions, through the membrane. Because most of the cell membrane is hydrophobic, these large charged molecules can't easily get through the cell membrane. So we add these openings that allow things to go in and out through the cell membrane through the proteins embedded in the cell membrane. Okay, so we can see this protein right here, right? This would be a transport protein. And you can see, right, it's an integral protein because it passes all the way through the cell membrane, and it's a type of transport protein because it can allow things to pass through the cell membrane. For example, water could use this protein to be transported into and out of the cell membrane. Okay, and the thing that can be passed through is determined by the shape of the protein, and the shape of the protein is determined by how it's folded when we look at its tertiary or quaternary structure, right? And this protein is going to be really interesting because the amino acids near water are going to be like polar and hydrophilic. Probably the amino acids in the core of the protein will be polar and hydrophilic. And then the amino acids that are close to these fatty acid tails will be hydrophobic, which is what allows that part of the protein to sit in that part of the membrane. Okay. Now, the next type of protein I want to talk about is a glycoprotein. A glycoprotein, okay, gets its name. The glyco part is in reference to a carbohydrate, and then protein. A glycoprotein is a protein with a carb attached to it. And this is usually for signaling or marking a cell. For example, this can help you determine if a cell is part of your body or not, right? And you can see here in our example, here is a glycoprotein. Here's the protein. This is a peripheral protein because it doesn't go all the way through the entire cell membrane. It kind of sits on this edge of the cell membrane. And it's got the protein part. And then on the top of it, it has a carbohydrate group. So that's why it's known as a glycoprotein. And this sits on the outside of the cell, and this can signal, hey, this specific carbohydrate says this cell is part of my body. Or it can say, hey, attach me to another cell. So these carbohydrates are attached to proteins here. And these carbohydrates are incredibly important because they act as signal molecules. They help identify cells. You on your cells have specific unique carbohydrate groups that identify your cells as your cells. And this is when we get into issues with organ transplantation and like not matching signals, which we'll talk about later. And carbohydrates can also hold cells together. So different cells can have different carbohydrates sticking to them. And then when the cells get near each other, the carbohydrates can kind of zip together like a zipper holding the two cells together. Okay, so that can be a part of a glycoprotein that forms a cell junction. Okay, so anyway, that's our carbohydrate right here. We talked about transport proteins and glycoproteins. We can also have enzymatic proteins. Enzymatic proteins can catalyze reactions. Okay, we have a lot of these in the mitochondria and in the chloroplast. And enzymatic proteins are usually peripheral proteins. They sit on the inside of the cell membrane. And they kind of like something might go in and then they make like a final product here, right? So they kind of sit on the inside of the cell membrane and help catalyze reactions. Okay. There can be free-floating enzymes too, but some enzymes are attached to the cell membrane. Cell junction proteins help hold cells together. Some of them fit together, like the proteins are like a lock and key and they fit together like two hands fitting together, like a lock and key to help keep the cells together. Some of them are glycoproteins, they zip together that way. And then this is kind of a really important one that we don't talk about in earlier biology classes, but in AP Bio, we spend a lot of time on this idea of cell communication, and it becomes really interesting. Proteins can receive messages on the cell membrane, right? And then relay that message inside the cell to get some sort of result, right? So if we look up in this picture, this isn't actually good because this looks like another transport protein. This looks like another glycoprotein. But you could have a protein that kind of like, I'm just adding another protein in here. Like let's say you had another protein in here and it got a message here. And then it encouraged, like passed on the message inside the cell so you could get a final result, right? So again, so proteins do all sorts of different things in our cells. Okay, I hope you enjoyed watching this introduction to biomembranes. Thank you so much.